Thanks, Nir. Thanks, everyone, for coming. A couple brief announcements before we get started. I wanted to make sure that everyone was aware that Thursday at the Delta Stewardship Council meeting, there will also be some data to decision-making subjects on their agenda. This Thursday, they'll hear from staff and panelists on the role of the Collaborative Science and Adaptive Management Program, colloquially known as CSAMP, and the Council will also hear some updates from the Delta Independent Science Board. Additionally, I wanted to make sure that everyone knew that this is the second of three presentations in a series. Do you have that on there? Okay. Thank you. There we go. The second of three seminars in this series. The next one after this one will be Tuesday, October 23rd. It will be from Louise Conrad. It will be on the second floor of this building, again, a brown bag at noon. That'll be titled Ecological Synthesis Within the IEP, Growing a Program to Make Long-Term Data Talk. So to today's presentation, Lauren Damon thought that it might be good to call out for you the amount of time that she spent in direct contact with the fish she is going to be discussing today. 7 years on some Delta Smelt surveys. She mentions that she used to handle smelt when they were more abundant than they are now, and so they had a lot more opportunity for hands-on experience. She feels as though the previous experience she had was influential in understanding the underpinnings of what makes an estuary work, and also she wanted to mention that she spent some time at the UC Davis Fish Conservation and Culture Lab, again a place where you come into rather intimate contact with Delta Smelt and other fishes under culture. So with that as background, I wanted to also mention that Lauren comes to us via the University of California, Santa Cruz, where she has a Bachelor of Science in Ecology and Evolution. Lauren's been part of the Interagency Ecological Program, or IEP, as a California Department of Fish and Wildlife employee since 2008. She's worked on Delta Smelt larval surveys, the spring Kodiak trawl, and the 20-millimeter survey. And just recently, Lauren took a promotion, changed roles within CDFW. She's now a senior environmental specialist working on voluntary agreements with water rights. But today she's going to be talking to us about her experience with Delta Smelt, their resilience, and their reproduction. So with that, thank you, Lauren.
Okay. Thank you, Steve, for that, and Nir, and thanks you guys for being here, and thanks to everybody also on the web who's watching this. I'm excited to talk about this with some work we did a couple years ago on Delta Smelt, fecundity, reproduction, and resilience. I'm going to talk about that today. So first, I just want to thank a couple people who are really awesome that contributed to this. First, Steve Slater and Jim Hobbs produced some figures for this talk and also provided some ideas to make it better. So some of this stuff that I'm going to talk about today also came from them. There's a couple of folks who listened to an earlier version of this talk and provided some feedback. Of course, I want to thank all the lab and field staff that helped with this special study that we conducted and just the monitoring surveys in general, and then all the people who have provided guidance, input, reviews, et cetera, on the work that we did here, including the manuscript that was published a couple years back. So for today's talk, I'm going to talk first about some basic Delta Smelt biology and history, and then we're going to go into a little bit about how they're monitored within the Delta, and then we'll talk about this special fecundity study that we conducted a number of years back, and then how I think that affects Delta Smelt's resilience and their management. Okay, so first Delta Smelt biology and history. The first fun fact is that Delta Smelt are in the family Osmeridae, and the root word osme is Greek for pungent smelt. And so if you've ever held a cucumber, I gave it away already. If you've ever held, I gave away the punchline. If you've ever held a Delta Smelt, either in culture or in the wild, you know that they smell like cucumber. So they're family name is very fitting for them. As adults, they reach about 2 to 3 inches, so they're not very large. You can see them pictured here on a measuring board that's in millimeters, so you can kind of get a reference. They're found in pelagic habitat, which means they're really just in open water. We don't find them near the shores or on the bottom. They're kind of out in open water. They're an annual species, so they complete their entire life cycle in 1 year from hatching to spawning, and then they feed on small invertebrates, typically zooplankton, but also sometimes mysid shrimps. Delta smelt are endemic to the San Francisco estuary, so this means that they're only found here, and this is the only place in the world where they can live. They can't go anywhere else. They evolved many, many years ago from a landlocked population of surf smelt, and so that's their closest relative. And because they're so intimately entwined with the system, because they evolved here and they're highly adapted to live here, they're really good at being an indicator of the status and health. So delta smelt are very adaptable to the estuary and the dynamicness of the estuary, but when the estuary isn't functioning very well, the delta smelt are functioning very well. So we use them as an indicator of the status and health of the estuary. So I mentioned they're an annual fish. This is their life cycle kind of on a linear timeline here. So you can see that adults are present in the estuary in the winter and early spring, and then they hatch out and larvae are present later in the spring, and then the juveniles and subadults rear over summer and later into the fall. So delta smelt have, they're an opportunistic strategist, which means their life history strategy is really to have a short generation time, and they produce a lot of offspring. So this is the conceptual model of their survival rate. This is from the Bennett 2005 paper, and so this is just so you can see that they produce a lot of eggs, and then there's pretty high mortality of the earlier life stages, and then you get left with only a couple of adults at maturity. So delta smelt experienced some pretty severe declines over the last few decades, and so they were listed both in California and federally as threatened, and then they were petitioned to be uplisted as endangered federally, but that was warranted but precluded. And so they're listed as endangered in California and threatened federally. This is the U.S. Fish and Wildlife Service critical habitat map, and so this is just showing that their critical habitat is essentially the California Delta. Water gets moved around California via 2 major water projects that are operated by a state agency and a federal agency. And so when delta smelt were listed, those water projects needed to get permitted under the Endangered Species Act. And so the U.S. Fish and Wildlife Service consulted with the Bureau and with the Department of Water Resources to permit their water export facilities that are located in the South Delta. And so they came up with some reasonable and prudent alternatives to minimize those export facilities' impact to delta smelt. And those RPAs specifically called out some specific monitoring surveys and data reporting in near real time to water managers so that water managers could adaptively manage delta smelt and the impacts of the water operations. So now we're going to talk about what those monitoring surveys are and just go into some basics about that. So the water projects were being developed in the 50s and the 60s. And so as a result of that, the Department of Fish and Wildlife and the Department of Water Resources got together and they agreed that there needed to be some studies in the Bay Delta region to understand really how these water projects were going to impact the ecology of the Delta. And so this was the early formation of the Interagency Ecological Program, was an agreement between these 2 agencies. It's now 9 agencies, but their mission statement is essentially the same, to use science to understand how the ecosystem is affected by the water projects and then how we can manage that better. So the first survey that got brought online by the IEP that's still running today is called the Summer Townet Survey, and it started in 1959. And you can see it operates over the summer, and it uses a rigid mouth, and it's mounted on skis, and the mesh size is such that it targets young of the year fish found in the estuary. It was originally intended for striped bass because that's what the department cared about in the 50s and 60s with striped bass. But it's actually proven to be really good at catching a variety of fish species throughout the summer that are present. So it uses it samples the entire water column using an oblique tow, and so what this means is that the net is let out to a specific depth depending on how deep the water is, and then over the course of about 10 minutes, The net is towed in an oblique direction towards the surface, so you can sample the entire water column. So there's going to be an animation of this next. It's sped up really, really fast. So imagine this over the course of 10 minutes. So the net gets put out first and then sets down, and then 10 minutes starts here, and then the 10-minute tow occurs like that. So you can get all of the fish sampled through the whole water column. Here's a map of the summer townet survey stations. This is just to show you guys that this survey samples pretty much the entire upper estuary. The black dots were the stations that have been around since the survey's inception, and then the colored dots are ones that have been added on later to increase their spatial coverage. So the second survey that came online was the Fallman Water Trawl in 1967, and it operates during the fall time period. It doesn't have a rigid mouth. It's closer to what you would consider like a traditional trawling net. The mouth is held open by the doors that you see dangling there. And the mesh size is quite a bit larger than the townet mesh, and this is because it targets a larger size life stage. Again, it was intended originally for striped bass because that's what we cared about, but it definitely samples a whole suite of species and is also towed obliquely. In addition to being called out in the biological opinion, this survey is used in the recovery plan for native fishes. So as things hopefully start to recover, this will be the survey that we use to track their recovery. Here's the station map for the Fallman Water Trawl. You can see the coverage extends quite a bit into San Pablo Bay, and then in recent years we've added quite a number of stations in the North Delta and the Central Delta to increase coverage, particularly for Delta smelt, but other species as well. So both of those surveys that I talked about are what we would call our longest running surveys, our longest long-term monitoring surveys, and so we calculate abundance indices for both of these surveys. So an abundance index is just to get an idea of the relative abundance of fish. It's not an absolute abundance, it's just to compare among years, statuses among years. And so both of these indices were used in the petitioning of Delta smelt when they were first listed in the 90s. And you can see, particularly in the summer townet survey is the one on the bottom, you can see pretty clearly the declines that have been experienced. And so these abundance indices are really good for monitoring trends like this over time. Okay, so after Delta smelt, or while Delta smelt were being listed, rather, there was a need for additional information about larval distribution and abundance, particularly in the South Delta where they were vulnerable to entrainment. And so the 20-millimeter survey got brought online in 1995 to operate during the springtime, and this one was specifically to target larval and juvenile Delta smelt around 20 millimeters in length, because that's when the projects need to start enumerating them for take purposes. And so managers really wanted to have a heads up about when larval and juvenile fish would be present in the South Delta to minimize their risk of entrainment there. They're not very good swimmers. So because the fish collected on this survey are so small, they usually range between like 3 millimeters and around about 40 to 45 millimeters, the entire sample gets brought back into the laboratory for processing. So this field survey in particular has a major laboratory component. And so here's a couple of the lab staff working up samples, picking the fish out of the debris, and then one of our larval fish ID experts identifying the larval fish under a microscope. So the 20-millimeter survey has a near real-time reporting requirement, and this is to give managers a real-time heads up about when and where the fish are occurring so that they can adaptively manage for them. So there's a website that's listed there at the bottom, and it has an interactive map that you can use, and you can select which species you want to look at. It doesn't have to be Delta smelt, it could be any species that we catch. And then the bubble plot will pop up showing you the catch per unit effort at each of the stations. The white circle means there's nothing there. The green ones mean that there was catch there. And of course the size is dependent on the CPUE, catch per unit effort. So sometimes if you look at the web page, because of the near real-time reporting requirement, these web pages get updated about once a week. And so sometimes partial surveys get updated, and this is so that managers have a constant stream of data available to them. So if you go to the web page and the map looks something like this, where you're like, that doesn't seem right, there's not really enough bubbles filled in there, up at the top where it says survey in progress, that's your hint to know that it's still not completely updated yet. And so as soon as that survey in progress disappears, then you'll know that that 1 week long survey has been completely processed. Okay, so here's just the recap of the I'm sorry, no, we're not there yet. So the last survey I want to talk about is the spring Kodiak trawl, the most important one for this talk. And that got brought online in 2002, and it really was an extension. There used to be a spring midwater trawl that sampled in the springtime, and so that ended up turning into the spring Kodiak trawl because the Kodiak trawl has improved detections of delta smelt. So they really wanted to improve detections, especially as delta smelt abundances started declining. They needed a way of more accurately sampling for them, especially in the South Delta. And so the spring Kodiak trawl got brought online in 2002. The survey, because it samples spawning adults, it provides water managers an opportunity to predict when spawning is going to be occurring, and the intent of that is to predict when larval fish will be present in the system. And so the spring Kodiak trawl then also has this goal of tracking spawning and spawning development and when and where spawning will be occurring or is already occurring. So the way that we do that, the way that we track spawning, is that all of the adult delta smelt that get captured in the spring Kodiak trawl survey are examined for their gonad development. So their gonads are their reproductive organs. And all of those get examined in the field using these tables similar to this, so the scientists on board can open them up and look inside and see what stage their gonads are in. The top picture is a male, and it's what we would consider about a stage 3, which is developing, or I'm sorry, stage 4, which is considered near ripe. It's almost ripe, but you can see there's still, the gonads are still relatively small. And same thing with the female. The gonads are still pretty small, but this is what we would consider a real near ripe female because the eggs and milt are starting to develop. So this is a ripe female, so she's in spawning condition. She's ready to spawn, and you can tell this because her eggs are large. They're fully yolked, which is why they're orange. And her ovary takes up pretty much her entire abdomen, and so you can tell that this is a ripe female. Probably if you put some gentle pressure on her abdomen, she would start releasing eggs, indicating that she's ready to spawn. But you'll notice that there's a couple of large discolored eggs up at the top of the photo, and these are eggs that are left over from a prior spawn, and so this means that this female actually has already spawned one time, so we would call her post-spawn or spent. Sometimes spent is used. She's already spawned once, and so she's actually a stage 6, meaning she's spawned already, and then she's developing this additional clutch of eggs that's ripe. So the spring Kodiak trawl has a web page as well, where you can, it's interactive, so you can see where the fish are, where they're caught, and you can also see, like this map shows here, the sex ratio, so the number of males and females captured at each site. And then you can drill down to and select, if you want to look at just females, you can see what phase of development they're in, or you can select just males and see what phases are caught and where. Okay, so here's our monitoring summary. So you can see which life stage of delta smelt each monitoring survey more or less targets. I use that term kind of loosely. And then the link down at the bottom, this is where you can access all of those survey web pages, and data is available. So last month, Vanessa talked about big data. All of these data are available online in raw form on an FTP site, including the metadata, just housed in databases. There's also the interactive web pages you can use, and then memos and such are provided in a bibliography. Okay, so now let's talk about the fecundity study. So while we were out on the spring Kodiak trawl, we made a lot of observations, obviously, about spawning, because that's what we're looking at is spawning. So while we were out on the spring Kodiak trawl, we kind of saw this opportunity that we're collecting data on length and we're collecting data on water quality, but we're also looking at gonads. And so if we took it one step further and we collected some data on their fecundity, we could put together this pretty comprehensive study about delta smelt reproduction. And so what we wanted to do was kind of define the length fecundity relation. In this study, we wanted to define the length fecundity relationship, and we wanted to see if we could put some observations around the time of year that they're spawning. So is there a spawning window that we could bound by, say, like temperature or size or something along those lines? And a couple other questions were our intent for this study. So UC Davis operates this fish conservation and culture lab, and they collect wild Delta smelt and then they spawn and rear them in captivity. And so this has provided an opportunity to observe spawning in captivity and some spawning behaviors. And so in communications with them and some observations that we made on the spring Kodiak trawl, we really wanted to see if wild Delta smelt behave similarly to how Delta smelt behave in captivity. And so in captivity, they're capable of producing a number of multiple clutches per year under controlled conditions. They have their own fecundity estimates, and it's very, very common for Delta smelt at the Fish Conservation and Culture Lab to live longer than 2 years. And so we wanted to see if we could identify if Delta smelt were doing the same thing in the wild. Excuse me. Okay, so our timeline that we were talking about earlier, the annual timeline for Delta smelt is up at the top, but this slide is really zoomed in on the spawning period, so we can talk about what FCCL observes during the spawning period and then define some terms and what we're trying to get at with the fecundity study. So adults typically are spawning between February and May. This is really variable, but they can spawn anytime between February and May typically. So if we're talking about the entire population, this would be the spawning season. But if we're talking about just one individual, this is a spawning window. And so it's typically bound by temperature. And so at the beginning of the spawning season, assuming a female is ready to spawn, she's large enough and her gonads are developed enough, a clutch can be released at the onset of spawning. And then there's some necessary time that needs to go by so that she can redevelop her gonads and divert energetic resources so that the eggs can be developed again. And so we call this time the spawning interval. And then after that spawning interval, she can hopefully release another clutch, and then another spawning interval, another clutch. And so depending on the length of the spawning window, you could have multiple clutches within that. The spawning interval in culture is around like 40 to 50 days, and those are held at constant conditions. So the spawning interval is probably based on temperature. The fish in culture are held at 12 degrees. But that's our best estimate so far of the spawning interval. It's about 40 to 50 days. And then the number of clutches a female can release in the spawning window is called the spawning frequency. So I'm going to use that term later. So it's just the number of clutches. So for the fecundity study, we took all the near-ripe females that we caught during the spring Kodiak trawl, and we opened them up in the lab, and we did egg counts on their near-ripe clutch. And so the eggs, we divided them into 3 phases. The developing phase is A, the A is pointing to in the picture. Those eggs are smaller and translucent. They don't have yolks developing yet, so those are what we would call developing eggs. B is the ripe or near ripe clutch, and so this is defined really by them having a yolk body that's fused together, and the egg is much larger. And then the post-spawn eggs we saw in the earlier picture, those are the eggs that are left over from the prior clutch. They're not pictured here, but those would be much larger, and they're usually just colored and kind of mottled looking. So for the fecundity study, we counted all of the eggs in the near ripe clutch because those are the ones that are the viable eggs that would contribute to the fecundity. So we did the study for 4 years, from 2012 to 2015, and we were able to look at 129 fish, and their mean fecundity was right around 2,000 eggs per female. So we plotted the fecundity as a function of fork length and found that there was no significant difference in the length fecundity relationship among years. And so this was one of our main findings, was that this relationship was consistent among years. But what did change, that you'll see in the table at the top, is the abundance. So we took we systematically subsampled the near-ripe females caught on the spring Kodiak trawl. So in 2012, there were 67 available to us, and by 2015, there was only 8 available for us to use. And so after combining, we just decided to group them all by year and then determine that the length fecundity relationship is a vital rate for Delta smelt. And this is also because in culture, the length fecundity relationship is essentially the same. The fish can get a lot larger in culture, so they have that 80 to 100 millimeter range filled in quite a bit more, but essentially this relationship is consistent for Delta smelt. Even though there is some variation, we can predict fecundity based on their fork length. Okay, another part of the study was to try to describe the spawning window bounds, and so we plotted the females that were in spawning conditions. So for us, that meant they were ripe or ready to spawn, and the post-spawn fish who have already spawned once. We just plotted the frequency of those and found that spawning typically occurs between 9 and 18 degrees. That's when we observed spawning to be happening. We also found that there was no smelt smaller than 55 millimeters in spawning conditions, so we said that that was the minimum size required for spawning. Another part of the study was that we looked at female growth, so if they continue to grow throughout the spawning season. The green box plots here are pre-spawning females, so ones that have not, or are not in spawning condition or have not spawned yet. And you can see that they do still grow throughout the spawning season. From January to May, growth is occurring even though they're currently spawning. So I think the motto is that it takes a year to make a smelt, and I would take that a step further and say it takes a year to make a smelt fecund, because they need that growth throughout the whole year. So this is a graphic from Jim Hobbs, and he plotted the age of the smelt with 0 being the day at hatching. So he plotted the age to the length, and you'll see that the larval and juvenile periods are really times for this rapid growth, that they spend these early life stages growing so that they can get as big as possible, hopefully so that they are fecund as adults. And so when they become subadults in the fall, energetically they shift and they switch to more gonad development. So growth slows during that time period. And so it's really the larval and juvenile stage that's important for ensuring that they're of adequate size during the spawning season. And so I just put a note here that growth during the entire life cycle determines their adult clutch fecundity, that the fecundity is about the adult life stage, but it takes a year to make a smelt. Okay, so we also wanted to see if 2-year-olds were contributing at all to the fecundity of the population, of the delta smelt population. 2-year-olds and 3-year-olds are common in culture, but we didn't know if they were common in the wild. And so this is all of the length of all of the individuals caught during the spring Kodiak trawl from 2002 until I think it went to about 2015. So you see that there's not a whole, the red triangles are what we thought were probably 2-year-olds, and not a whole lot of 2-year-olds present in the population in the wild. We don't know if they historically were more abundant or not, but because 2-year-olds are disproportionately more fecund than 1-year-olds, it might be a benefit to have additional 2-year-olds available for spawning purposes because they contribute so many more eggs than 1-year-olds. Okay, so like all good studies, we needed a conceptual model to try to get a handle on what parts contributed to the annual fecundity. So really what, we care about the clutch fecundity, but really what we want to know is how many eggs can each smelt produce in a year. And so that's the annual fecundity. And so this is just breaking down the parts of annual fecundity. So it's really dependent on the clutch fecundity, how many eggs they produce in a given clutch, and the spawning frequency, how many clutches they can produce. And so essentially this is dependent on their size, which is dependent on how much food and resources they have available to them for their whole life. And then the water temperature really dictates when spawning starts and ends, how long the spawning interval is, and that determines how many times they can spawn. So if you want to boost up the annual fecundity of a delta smelt, you want to make sure she's large, so her clutch fecundity is high, and she has many opportunities to spawn so that the spawning frequency is high. Okay. So now let's talk about what I think this means for delta smelt resilience and management. So when you're thinking about resiliency in terms of reproduction, you really want to talk about their reproductive potential. What is their potential to reproduce? And it's their capacity to produce eggs, essentially. How many eggs can they produce in a year? And so it's their annual fecundity that's possible. Because theoretically, the more offspring they can produce, the more resilient they will be later on. So Steve Slater developed this conceptual model for delta smelt that shows what the reproductive potential could be. This is just a theoretical model, so these numbers aren't real. And he plotted a theoretical previous year class abundance, applied a mortality, a survivor curve to that, and then, so on the x-axis is month, and so it starts in April, and then it's a 2-year timeline. So you have this abundance, you apply a survivorship curve, and then it leaves you with the number of adults available for spawning, which you'll see is really low in this figure. And then for every clutch they can produce, their reproductive potential increases quite a bit. And then you apply the survivorship curve. And so what you could do with this is use it as a tool. If you carried it out in another couple of years, you can use it as a tool to predict how many offspring that you need either to increase your population, decrease your population, or maintain your population. So you can really know how many eggs you need to produce every year to get the certain number of adults that you want. And the good thing about this tool, which you'll see more later, is that you don't need any more additional monitoring than what we already have available to us. And I'll break that down and unpack it a little bit for you. But it doesn't require any additional monitoring, just some analytics. Okay, so to model the reproductive potential, we just go back to our conceptual model and see that if reproductive potential is really just a surrogate, or if annual fecundity is our surrogate for reproductive potential, then you need clutch size, clutch fecundity, so you need female size, and you need to know the spawning frequency, which is based on the spawning interval and the spawning window. So we already have female size because the spring Kodiak trawl collects adult females, and so we have their lengths available to us. The spawning interval we have estimated from culture. I said it was 40 to 50 days on average. So really what we need to know every year to determine annual fecundity is the spawning window. So you can do that two ways. One way is to just look straight at water temperature, and so water temperature available freely on sea deck. The issue with this is that it's highly variable, so applying the assumptions and saying this is the time when water exceeds 9 degrees can be a little bit of a challenge, but it's possible. The other way is to use survey detections, and so that's what we did for this study, was we bound the onset of spawning as when we first saw fish in spawning condition during the spring Kodiak trawl, and we bound the end of the spawning window by when we last caught newly hatched larvae in the 20-millimeter survey. One of the caveats to this is that, especially when abundances are low, just because you don't catch them doesn't mean they're not there. So that's just something to keep in mind, that these are just estimates and probably they can be improved upon at some point, but this is kind of what we've got available so far. So we looked at spring Kodiak trawl data. Spring Kodiak trawl is also a monthly survey, which I didn't mention, but it's a monthly survey, so the resolution isn't great. But we looked, this is 2012 for an example, so if we wanted to bound the spawning window, you can look and see when you first collect female delta smelt in spawning conditions. So this is February of 2012. We saw post-spawn fish and ripe fish. So we said that February of 2012, about the middle of February, was when spawning started. And then the figure on the right is a length frequency diagram taken from the 20-millimeter web page. The x-axis is length, the y-axis is frequency, and then each one of those individual graphs is a week-long survey. So it's chronological survey 1 at the top, and then it goes down to survey 9 at the bottom, and so these are the 9 week-long surveys that occur during the 20-millimeter survey season. And the box that I've highlighted there are fish that are 5, 6, and 7 millimeters, and so that's what I'm considering newly hatched larvae. And so you'll see that in the middle around survey 7, I can use this to show you guys. This survey here is the last time, or actually survey 6, was the last time we saw these newly hatched larvae present. So if you back calculate a couple of weeks for incubation time, that would put the end of spawning occurring somewhere around the beginning of May. That makes sense. So for 2012, using this method, we bound the spawning window between about the middle of February and about the middle of May. So then you can plot that. This is day of year on the y-axis and year on the bottom. This only went through 2016. But you can see the length of the spawning window for all years. So we did that similar method for all years. And you can see the green bars are the length of the spawning window. And then the black bars are spawning events. So if you assume that they spawn on the very first day of the spawning window and you went 50 days later, that's where your second black bar is, and another 50 days later is your third black bar. And so you can figure out how many spawning events are possible each year. And that's what the table over on the right is the number of spawning events that we calculated based on certain parameters, or based on the spawning window that we calculated. So this is highly variable, right? Every year is different. Some years you maybe get one spawning event, like 2009, that looks like maybe there's only one spawning event, whereas in 2010 and 2006, you could easily get 3 spawning events in there. And I will caveat this to say that not all the females are ready to go on the first day. So this would be best case scenario. Some of them probably need a little bit more time. They're not going to start on the very first day that the spawning window opens, quote unquote. And so not all females are going to be able to get 3, but best case scenario is they could get 3 or more in some years. So using all this information, we developed an egg production index that's really a surrogate for reproductive potential. We're calculating the number of eggs that are possible per year. So we use the clutch fecundity based on length of females that were caught in February of the spring Kodiak trawl, added all those up, multiplied it by the spawning frequency for that year, and then ended up with an egg production index. So that looks like this for 2012 to 2017, and so this is really an abundance, a relative abundance of eggs capable of being produced per year. So this is Delta Smelt's reproductive potential per year. And so you can see that things change quite a bit. 13, 14, 15, and 16, you can see that the egg production declined quite a bit. But you might be wondering, what is this good for? What does this mean? Well, we thought maybe it could be used to predict a larval index. So if you know how many eggs you could potentially collect, could you then predict larval abundances using this information? Could you predict larval production based on egg production? And we think that yes. So this is the 20 millimeter index plotted with the egg production index, and they're really highly correlated. The trends all kind of match up. And so basically what this is telling us is that using spring Kodiak trawl data, you can predict what the larval production will be of the following year. Okay, so to wrap this up and summarize, so if you're talking about an individual female, her annual fecundity could be, so if you wanted to increase the annual fecundity of a female, you'd want to increase her size. You want to make sure that she's able to grow throughout her entire life cycle, especially during the spawning season, and that there's opportunity for multiple clutches. If you're talking about the population and you want to increase the annual fecundity and the reproductive potential of the population, that's really going to require some increased egg production for all of the individuals in the population, which means a longer spawning season overall so that all of the individuals can benefit from increased clutches. And you want to make sure that the females are large and can have adequate growth throughout their life cycle. So growth and survival are important all year. This really means that cooler temperatures for survival, especially during the summertime, when temperatures typically exceed their tolerances, not typically, recently exceed their tolerances, and of course food availability for growth, because we need the fish to be able to grow as large as possible so that their fecundity is as much as possible. So what I think the management implications of this are the things that we could do to really help Delta Smelt be as fecund as possible and improve their reproductive potential would be to provide them some more food. So frequent floodplain inundation to exchange the nutrients, habitat restoration so that you have productive environments for food to grow, and then suppressing clams, because if we have productive environments and invasive species are just eating the food, then that's not being used for Delta Smelt. And an issue in recent years has been water temperatures, so the spawning window kind of gets cut short because temperatures warm up in the spring and the summer, and so the spawning window gets cut short when temperature warms up later in the spring. And so there was a modeling paper put out in 2017 by Julia Vroom et al., and they were linking water temperature with flow. So really improving flows throughout that, and it was specific to the estuary. So improving flows throughout the estuary will help keep springtime temperatures low by decreasing the residence time, and then also the cool water releases from the reservoirs will keep the estuary cooler to provide spawning opportunities for smelt in the spring. So a couple of ideas for future work, places where we might want to consider refining this would be looking more at the spawning interval in the wild. The best estimate of the spawning interval, the time between spawnings, we have from culture. So trying to do some studies to see if we can get a handle on that in the wild and how it varies based on size and temperature. Refining this egg production model so that we can use it as a predictor tool. I think this could be a really powerful tool if we did some refinements to actually see how it could be applied in the wild as a predictor of the offspring needed to improve populations. And then my invitation to everybody to start getting creative about how we can improve food and temperature conditions in the estuary. And with that, thank you guys very much.
Thank you for the great talk. Are there any questions?
Can you give a little more your thought about the, especially in the drought and the, in drought condition in certain situations, the impact is on flow and also temperature. So what do you think about the management action for resilience for the long-term management of the Delta smelt?
You mean with like the drought in particular?
Yeah. That's the stress condition that might, will put a very big stress on the population, will be very important to address.
Yeah, so, in 2014, 15, and 16, that spawning window was really short based on what we saw likely because of the drought. And so I think there is opportunity here. I think dealing with temperature is going to be hard because it's related to flow and ambient air temperature, so that's a challenge, yeah. Getting cooler temperatures throughout the spring will really help with Delta smelt resiliency.
So you have that culture lab or something, that way you can back up and do something drinking with traffic. And population is very low, and I don't know.
So they have a genetic refuge population that's kind of there for that purpose. I don't know if they're talking about releasing fish from that facility yet. That's not my area, but it is available. There is a genetic refuge population at the culture lab. Mm-hmm. Yeah, they genetically manage that population to preserve genetic diversity. So yeah, that's what it's for. Mm-hmm.
I've heard that there are concerns about the midwater trawl, both the size of the openings in the net and where they survey are both problematic, as in the fish maybe are more on the edges rather than in the middle of the water body. How do you respond to those concerns? And the second question is, if this work was done post the special pod studies, correct?
Yeah.
Is there any work to go back and revisit those about 10-year-old studies now in light of this and other newer science, and say, how would the pod studies have looked if we had these advances that we've made here and elsewhere?
So I'll answer that question first. I wasn't around for the pod studies, so I'm not as familiar with what they were. So I can't really speak to that because I don't know how this would have impacted that. But the Midwater Trawl Survey, it's really about like long-term trends. So if you're looking at the entire life, so that survey started in 1967. So if you're looking at the whole time span, you're just really comparing among years. So if they were in different habitats, if they're in different habitats now, if they're not in a pelagic open water habitat now, then they probably weren't then either. So you're really just looking at the trends over time in that survey, and that's decreased. Whether or not there are other places, I think we have other monitoring surveys that handle that. There is beach stain survey data that's available. But the decline is pretty clear in the midwater trawl, and it's just about comparing among years in a habitat that we've been sampling long-term. Yeah.
Hi, thanks, Lauren. You had a slide where you showed there were two methods that were possible to examine some of the parameters of the spawning window. There it is. And the one you went into detail was the second one, which you, as a member of the survey crews, would likely hit upon, and you make a pretty compelling case for what those spawning windows are and how they change over years. I'm more familiar with the sort of temperature-related models and some of the recent otolith work that back calculates growth parameters based on temperature, and so the first method for sort of getting at that. And I'm broadly of the impression that those methods, while quite different, actually hit upon essentially the same conclusion. And I didn't know if I could check with you whether or not my impression about those two methods are correct, whether they're broadly comparable, meaning you've got two pretty different methods that end up with pretty similar conclusions, which would lead me to believe that, you know, the overall concept that you have is robust, and your conclusions could be, you know, carried forward.
Yeah, I think with the water temperature, the issue that I looked at CDEC data and did this with temperature, and it's hard to put a time on like when you surpass 9 degrees, because in Rio Vista it's different than it is in Suisun Bay. And so I think to use that method, some people would just have to agree on what station we're using or what average stations we're using, and just decide how we're going to interpret when we surpass 9 degrees and when we surpass 18. But yeah, essentially they get at the same thing, and they're not super far off from each other. There is an issue of earlier in the season, in most years the water temperature is 9 degrees, like it doesn't really ever get below 9 degrees. So that was kind of a confounding factor as well, is that you don't ever really get below the 9 degree threshold in some years. So it's hard to, you know, it's hard to put a start with that. But I think ideally, you know, if this method were to be used for like predicting resiliency or trying to get at recovery, I think water temperature might be the way you'd want to go. You just have to figure out how you're going to deal with a couple of issues. Yeah.
Yeah, so the slide you had with the green bars, the spawning events, so I'm kind of curious if you know anything about what delta smelt do when they're spawning outside of their preferred temperature range. And I say that because in every year, delta smelt need to spawn, right? They're an annual species. So if the spawning window is really constrained in 1 year, they're still going to spawn anyway. And do you have any insight on probably what's going on when that happens?
Well, one, I think they're adaptable. So I think that they can probably make do with some less than great conditions in some years. You know, they're going to find, they can find a little refuge somewhere, you know, that places that they can use. So I think that's part of it, is that even though, you know, we're not detecting them in the survey data, or the temperature says it's been over a certain amount for however long, you know, there's going to be holes they can probably go in to spawn. It's just, when you're looking at that, you're talking about like maybe a couple of individuals as compared to like this whole population. And so what you really want is like things to be good for the whole population, not just a couple of individuals. You don't want to like push them so far to the fringes that they have no resiliency left.
I might have seen one question online, if you don't mind me. Earlier, you wrote that higher temperature were preferable for egg production, but your later slide called for lower temperatures.
I don't know. Did I say that? I don't know where he means I said that it was preferable for egg production.
Any other questions?
Help me.
Or maybe
Did I say that?
I don't think.
It may have been when you wrote. Yeah, I think it was a relative term of warmer temperatures, not lower.
Here? I think I just meant that temperatures need to be within the range of adequacy for growth and survival. I didn't mean it, we wanted it hot. I think just at the adequate range within the estuary. Yeah, sorry.
That's it. Thanks for the clarification.
Email me and I'll try to help.
I'm guessing you did this, but in your same green bar slide, did you go back and then compare that to the actual temperatures? Does it correlate the end of the spawning window?
I didn't look at the two, like I didn't really like check a correlation, but I did that analysis and I can show you guys if you're curious. Somewhere, oh, here it is. Yeah, and so really what you get is just a lot of years where the 9 degrees, you never go below 9 degrees. And so it made the season really long in a lot of years. So I arbitrarily cut it off at 30 days because we don't really see spawning occurring in January. So I arbitrarily said that February 1st is the earliest. And so in a lot of years, you hit 9 degrees before February 1st. And so you couldn't really see as much of a trend because it made the windows a lot longer. So I think there's more in play than just the 9 degrees, I guess is my point. Yeah.
Yeah, those are bigger than what you.
Yeah. 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 And so I think that also goes back to if you were going to do the temperature route, we just need to figure out what parameters we're going to put on that and what stations. This is at Rio Vista. I looked at water temperature. And so you probably want to use some sort of average, like across the estuary. You probably wouldn't want to just pick one, especially one in a river area, because these did make it big. And then this didn't correlate to the 20 millimeter index. There was no correlation. But this, I think, is what you were getting at, the hatch dates, spawning windows. So this is hatch dates from Jim Hobbs, and so these more closely matched what we did with the green bars.
I have 2 questions. One is you mentioned clam suppression.
Yeah.
Are you aware of any work that's going on in that area? And second is relative to cooler water in the Delta. You mentioned that there's a study that shows more flow and higher releases is cooler water, but is that in a quote-unquote normal year, or is that in a real wet year that occurs? I mean, because I've never heard that, like, you can up releases in Folsom and cool down the Delta. I've never heard anybody say that, that it's so impacted by ambient air that that's the driver.
Yeah. I don't know anything about clams, unfortunately. I don't know. I'm hoping there's studies being done, but I don't know any of the active stuff happening right now with clam suppression. And the study that I was referring to, the lights are on now, but it's at the bottom there was the reference. And I think what they did was modeled temperatures throughout the estuary for a number of years to see, to change some parameters and see if they could be predictive of temperature. And where flow helped reduce temperatures was when you decrease the residence time on like floodplains and stuff. So by decreasing the time that water just sits there so the ambient air temperature can affect it, it's really more about just kind of moving that water out, and so it can't absorb the ambient air temperature.
So that's a great contradiction, right? So we, on one hand, we say we want water on the floodplain to produce food, but now you're saying maybe that's a bad thing for temperature. Now, and also when you look at the whole flow question, the conflict between winter run and temperature control management at Shasta and flow is it's hard to work inside those conflicts.
Yeah, I think with the residence time and with the floodplain inundation, it's really about getting the timing right. And so I think there's like a time that is good. Like we want the exchange of nutrients and we want the productivity, but you don't want the water sitting on there for weeks on end in the summer so it doesn't get super hot. So yeah, I think there is going to be, they sound like they're contradictory, but I think it's really about the amount of time that they're sitting there. You know, you want enough for inundation, but not so much that it's like a shallow marsh permanently.
Great. Thank you so much.